All right, good morning, everybody. I see several of you have figured out how to do the coloring. It's one of those things that when you first try and do it, it's really tricky, but you can get the hang of it. You get better at it. I hope you're enjoying doing this as we're waiting for everybody to show up. Unfortunately, I have to erase all of this in order to start showing the presentation. Oh my goodness, a lot of people are showing up late. Oh, actually, okay. All right. So as usual, we're starting with announcements. And as usual, we are going to have a quiz and a homework assignment that are both going to be due on Sunday, February 6th. This week, we're going to finish talking about nucleic acids, and we're going to talk about cells. And then next Monday, we have our first exam. I'm going to spend some time talking about that in just a second. But then next Wednesday, February 9th, we're changing topics and we're going to start talking about ecology. So if you are the kind of person who doesn't necessarily enjoy cells, we're almost done with them. So we'll be able to talk about stuff that most students find more interesting very soon. Okay. This is the first exam coming up. I've given you quizzes and homework questions to kind of, one of the reasons for that is to help you figure out how I ask questions. So there is a page under this week's module that talks about exam 1. We are going to have a review session on Friday. It will be recorded and posted to YouTube. Exam 1 covers material from the beginning of the semester through the end of Wednesday's lecture. It will be on Canvas. It will be available definitely Saturday, February 5th, and it is due February 7th before midnight. There is no lecture on February 7th. I will be on Zoom during class time, so if you read a question and you don't understand it, pop over to Zoom. I might be doing something else, so you're going to have to speak up and say you're here, you have a question, and I will be happy to explain questions or, try and help clarify questions if you have those problems. The exam is mixed format, so there will be around 30 questions. It will consist of multiple choice, true, false, short answer, and it will be timed. So make sure you have the time to complete it before you sit down. The exam is open note. Now, in my experience, students, frankly, they do about the same on an open note test as they do on a study for it test. Some students do worse. It sounds counterintuitive, but what happens is students do nothing to prepare for a test that is open note. So they think, well, I just read the notes. But they tend to run out of time. It tends to take longer to find an answer in your notes than you think. So there's information under this week's module that talks about that. I recommend either spending some time studying or organizing your notes. don't just highlight everything. It's very tempting to just highlight everything. But when I was in graduate school, taking open note exams, which are much, much longer than the exams you'll be taking, I created an index so that I could find everything. And when I say there was a lot more information, I would have a 2-inch, 3-ring binder for every exam. So I had to have an index if I was going to find stuff in there. All right. The project proposal was due yesterday. I have graded all of those. If you got a 2 on your project, you're good. Just go ahead and do it. If you got a 0 or a 1, read the comments on your project proposal. If you got a 0, you will need to resubmit. If you got a 1, you might need to resubmit. I might have mentioned that the topic needs to be refined, for example. If you have not submitted your proposal, you do still need to do that. The project is 10% of your grade. So it's a big, big deal. Now it's not due till March 27th, which is the Sunday before spring break. If you are doing a presentation, after exam 2, I will be sending out emails to schedule when to do the presentation. Or actually, we'll be in person by then, so I can just talk to you at class. And unfortunately, I have one more thing I have to talk about before we get started. I have found a couple of cases where people have been plagiarizing answers in their online assignments. Please don't do this. It is disrespectful to your fellow students. It's disrespectful to me. And frankly, everything online is open note. You don't need to plagiarize anything. You just need to look at your notes. So let me be very clear. If you copy something from another location, another person's work, and submit it as your work, you will get a 0 on that assignment that cannot be dropped. In addition, some people are posting questions on online forums and then putting the answers as their own answers. That is plagiarism too. One of the ways I can tell when this happens is that people start talking about something that is beyond the scope of the class. If you're talking about something that we have not covered in lecture and you don't list a source of that information, you will get a 0 on that assignment that you cannot drop. So be very, very careful about this. Okay. If you have questions about any of that, send me emails. If you have anything specific you want to talk about in the review session, send me emails about that. I'm not just going to sit here and run my mouth. It's going to be a question and answer kind of thing. So you need to have questions for me to answer, so I know what we need to go over again. All right. We have to talk a little bit about nucleic acids. This is a biological molecule or a macromolecule made of nucleotides. And most of this is going to be DNA, RNA, ATP, but there are a few other small molecules that are made of nucleotides. So a nucleotide looks a lot like what you see here. There is a sugar and a nitrogen containing base attached to it. This particular one is ATP. Here are the 3 phosphate groups on ATP. There's the adenine that gives ATP the A. So adenosine triphosphate comes from the adenine and 3 phosphate groups. DNA, of course, that's our genetic information. And it forms this double helix structure. And RNA, we're going to talk about what RNA does, but it's related to DNA, but slightly different. We're going to talk in more detail on that in just a second. We're going to start with DNA. Deoxyribonucleic acid. Deoxyribose is a monosaccharide. So it is a simple sugar. So here's a sugar. The nucleotide also has a nitrogen containing base and a phosphate group. The phosphate group and the sugar make up the sides of the DNA molecule, what we call the backbone. Or if you think of this as a spiral staircase or a twisted ladder, it's the sides of the ladder or the sides of the staircase. The phosphate group of one nucleotide is attached to the sugar group of the next nucleotide. So you end up with alternating sugars and phosphates along that backbone. There are 4 bases. Looks like I wrote over this one at some point. So let me add that there. There's adenine, thymine, cytosine, and guanine. Generally, when we start talking genetics, we will abbreviate these A, T, C, and G. The bases bind to each other, acting like the rungs or the steps on the ladder or staircase. The order of the bases is the genetic information. And that, of course, means that the order of the bases make up the the genes. These, of course, are hereditary. They get passed from parent to offspring. We're going to talk a lot more about that when we talk about genetics later on in the semester. Now, one thing that's very important to notice here adenine always binds to thymine. It never binds to cytosine or guanine. I keep this in mind because adenine and thymine, the letters that make up I'm sorry, the shape of the first letter in adenine and thymine, they're all straight lines. Cytosine and guanine only bind to each other. The first letter in cytosine, the first letter in guanine, both are curved. So that's how I can remember which one binds to which. And that's really an important thing. If you go on and you learn more about genetics or you learn more about how cells function, this is important to remember. Apples in trees and cars in garages. Yep, that's another way to remember it. Absolutely. If you go on the internet, there's probably a dirty one that will help you remember it too. I find that the dirty ones tend to be the ones people remember. All right. RNA. RNA is ribonucleic acid. So ribose is the monosaccharide here. So we've got ribose for RNA, deoxyribose for DNA. D means without or missing. Oxy refers to oxygen. So deoxyribose is simply ribose that is missing an oxygen. Tells you what it is in the name. RNA is organized similarly to DNA. There is a backbone made of alternating sugar phosphate groups. And there are bases that contain nitrogen. And RNA bases, you'll see cytosine, guanine, and adenine, just like DNA. But instead of thymine, RNA uses the base uracil. So that is one difference between DNA and RNA. While DNA is a double-stranded molecule that always forms a double helix, RNA is a single strand and it can have a lot of different shapes. So it can function kind of as a strand. It can form these really funky looking cloverleaf shapes. So lots of different shapes for RNA. RNA also has a lot of different functions. When you take biology and you learn about RNA, you learn about its role in protein manufacturing. But it also is important for gene regulation. It's important for cellular immune defense. So it can do lots and lots of different things, not just protein synthesis. And RNA is significantly less stable than DNA. So it won't survive as long as a strand of DNA. All right. So let's see how much you picked up from talking about DNA and RNA. Again, this is an anonymous poll, so feel free to put your answer down even if you're not confident in it. I'll give you 1 to 2 minutes. depending on how fast you guys answer. Okay, looks like probably everyone who's gonna vote has voted. So which base pairs with cytosine and RNA? Half of you got it right. It's guanine. Cars in garages, remember? The rest, not so great. This is an important thing to remember. Adenine binds to thymine. Adenine can also bind to uracil. Cytosine can bind to guanine. That's it. That's the only way this works. So remember that. Very, very important. Big hint about, the kind of question I might ask on the exam. Which of these are different between RNA and DNA? Good. Guys did pretty good here. Number of strands, uracil instead of thymine, ribose sugar instead of deoxyribose sugar. Those three are all correct. The last of the biological macromolecules that I want to talk about is ATP. Adenosine triphosphate. We will talk about ATP when we talk about photosynthesis, and I'm sure we're talking cellular respiration. So that's later on in the semester, but it's really important. It's an important molecule for cell function. ATP is cellular energy. And here's your adenine, your ribose, and 3 phosphate groups. So that's a phosphate group. That's a phosphate group. Oop, I gotta move that one over. It won't let me move it. Of course not. Hang on a sec. And that's a phosphate group. So you can see the phosphate groups share an oxygen between two of them. These phosphate groups line up, and the energy is actually in the bond between the phosphate groups. So that's the high energy bond right there. And that bond breaks to release energy to power other cellular processes. When that happens, ATP becomes a molecule called ADP and energy plus a phosphate group. So that phosphate group leaves. When it leaves, that energy is released. ADP, adenosine diphosphate. So there are two phosphate groups there. ATP is a relatively short-lived molecule in the cells. It'll break down kind of spontaneously in the cell. So you can't use this for energy storage. You just use it for short-term energy needs of the cell. And that's biological molecules. We're going to see DNA again. We're going to see ATP again. We're going to talk about proteins again. So don't feel like you can just shove this aside and not worry about it. We're going to come back and use this stuff later on in the semester. Anything you want me to go over about biological molecules before we move on and talk about cells? Yeah, so that may not have been clear on the screen. Let me type that again and make sure it's completely understandable. ATP breaks down to ADP plus energy plus a phosphate group. And when the cell needs to make new ATP, it starts with ADP, adds energy and a phosphate to become ATP. So the process goes in the ATP to ADP direction to release energy, and then to make energy that the cell can use from food, for example, it has to go the opposite direction. That's a pretty complex process. And we'll talk about it, but not till after ecology. So we'll get to talk about ecology before we get to that. All right. The cell. Have I shown you one of these pictures before? I know I've shown it to some classes this semester. If I haven't shown it to you guys, this is a false color electron microscope picture of the inside of a cell. I'm seeing lots of people saying they haven't seen this. Great. When we talk about the cell, we'll talk about mitochondria, and we'll talk about vacuoles, and we'll talk about the cytoskeleton. And most of the images are going to be very simplistic drawings of a cell. This is an actual image of a cell. This is a mitochondria. So this is not a whole cell. This is just part of it. So this is a lipid bilayer membrane. There is a second membrane in the mitochondria. And in that inner membrane, you can see these purple and yellow things here in the membrane. Those are proteins that are embedded in that membrane. I can't identify most of these, but I can identify this one. This is one called ATPase. It's one that makes ATP. How's that for cool? You can actually see it in this image. These things, that's part of the cytoskeleton. This is a vesicle. Vesicles carry materials around the cell. These are also cytoskeleton. All of these other things, these are all proteins, various different kinds of proteins. Now, I'm showing you this not because I'm going to ask you to identify anything from an image like this, but so that you can grasp that the cell is jam-packed full of stuff. The way we talk about it, we take these different components one at a time and talk about each, but they're all interacting with each other. And there are thousands of copies of all of the different proteins and thousands of copies of mitochondria, thousands of mitochondria in every single cell in your body. It's something that we often misunderstand when we talk about cell biology. All right. So why the hell do we need to learn about cells? Well, it's because of cell theory. Cells are the basic unit of living things. So they're the basic structure of your body. Your body is made up of trillions of cells. Everything that's living is made up of one or more cells. Our definition of life says that you got to have a cell. If you don't have a cell, you're not considered living. And the last part of cell theory, cells only come from pre-existing cells. So that's the only way you get new cells, is when cells divide. So here it is again, exactly what I typed up. The cells are the basic unit of the structure. That should say organization. I often get organisms and organization, and you can guess the other org word that I get confused a lot. All right. Now, the first thing I want to talk about is cell size. And so this image is going to talk about cell size. Most cells are microscopic. There seems to be a limit to the maximum size a cell can be. So eggs and a few other types of cells can be seen with the naked eye. They're still very, very small, but they can be seen with the naked eye. the limitation to size is because of this concept called surface area to volume ratio. So I'm going to go through this on this slide, and I'm going to go through it a second time on the whiteboard. So if you get lost, I'm going to repeat myself. The idea here is that the amount of surface area on a cell, the amount of membrane wrapped around that cell, limits the amount of nutrition, gas exchange, material that can be imported, and waste that can be exported. And a certain volume of cell requires a certain amount of gas exchange, import of nutrition, export of waste products. And as the size of the cell increases, the relative amount of surface area cannot keep up. So this graph, there's the radius of a cell. That's this dimension from the edge to the center. And then on this axis, there's the surface area to volume ratio. So you can see that small cells have proportionally more surface area. And once they get too large, the relative amount of surface area decreases drastically. So I'm going to go to the whiteboard and explain this again, because it's a complicated concept. So the idea here is that a given volume needs a specific amount of resources. Resources enter cells across the membrane. So all cells are wrapped in a membrane. The surface area limits the amount of resources that can enter a cell. Those resources can be nutrition, material for building macromolecules. It can be waste exiting the cell. It can be oxygen entering and carbon dioxide leaving. All of these things have to cross a membrane. So when we look at this, I'm going to walk you through this process. We're going to describe it based on the size of based on a cube, even though cells are not, well, some cells are cube shaped, but usually not. The math is just easier. So if we have a cube that is 2 millimeters on each side, or centimeters or inches, or whatever unit you want to talk about, the amount of total surface area of that cube is 24. So let's say that's square millimeters. And the volume of that is going to be 8. It's going to be 8 microliters, 1 cubic. Yep, 8 microliters. I think that's right. Anyway, the ratio of the surface area to volume, 24:8, or in simple terms, simplified, it's 3:1. Now let's double the size of each side of the cube. And when we do that, the surface area goes from 24 to 96. That's a big jump. The volume goes from 8 to 64. So that's 96 to 64, or 3:2. So we can already see that there's a big shift in how much volume there is compared to the amount of surface area. If we do 12 by 12, oops, surface area is 864. Volume is 1,728. And right away, you should notice now the volume is a lot more than the surface area. It's a much bigger number. So we're going to start having problems getting material across that surface to supply that much volume. So how are we doing on this? This is complicated, and I want to make sure you guys are following this. One of the things this means is that a large number of smaller cells can do more work than a single small cell. Simply because a bunch of smaller cells that have the same volume as one large cell will have a lot more surface area. So they can take in more nutrition and molecules and oxygen. Okay, so that's surface area to volume. I hope you guys got that. Cells aren't always round. So all that stuff I talked about, surface area to volume ratio, when we looked at round and square type cells, well, this is a cell kind of elongated. First off, that gives you more surface area for the amount of volume. So cells like this nerve cell that have all of this surface area and a relatively small amount of volume, they can be a lot bigger than something like an epithelial cell, which do tend to be kind of square shaped, cubes or rectangles. Skeletal muscles tend to be very long and thin. Sperm, of course, very complex shape. This is an immune system cell. It's kind of amoeboid, like an amoeba. It's got all these projections. Folds and extensions of the cell membrane can increase surface area for a better surface area to volume ratio. All cells must be able to do certain functions. They must have genetic material. I'm putting an asterisk here. They have to have genetic material during at least some part of their life. If we look at human red blood cells, when they are fully mature, do not have genetic material. So there are some exceptions. The human red blood cells have genetic material early in their development process, but they lose it later on. Cells must be able to regulate the material that enters and leaves the cell. Got to have some control over that. You don't want just anything getting in. They must be able to convert food to cellular energy. That's where ATP comes in. That ATP is our cellular energy. And they must be able to manufacture carbohydrates, proteins, and lipids as needed. I'm sorry, I'm getting distracted by the trash guys outside. Their truck has trouble turning around at the end of the cul-de-sac. It's always funny to watch them trying to figure out how to turn that thing around. And that rambling was so you guys would have time to write this down. So these are the basic functions of all cells. Okay. When we talk about types of cells, we're going to talk about the difference between prokaryotic and eukaryotic cells. So prokaryotic cells, you've probably heard these terms before. Prokaryotic, notice I spelled it differently here. I spelled it with a K in one place and a C in the other. You'll see it both ways. K seems to be the more common spelling these days, but sometimes you'll see it with a C. So prokaryotic, we break this word down. Pro is before, karyon refers to the nucleus. These cells evolved before the nucleus evolved. They're older cell type. They still exist today in bacteria and in a group called archaea. And they're small cells. They lack that nucleus. They don't form complex organisms. They tend to be single cells, although they can form clumps, colonies, and aggregations, which are multicellular, but they don't specialize different cells for different functions. They don't have a complex body like we do. or plants or other animals. Eukaryotic cells, on the other hand, these are much more complicated. EU means true. So anytime you see that in a scientific word, that means true. And karyon, again, is nucleus. So eukaryotic cells have a true nucleus. And that's the defining difference between these cell types: the presence or absence of a nucleus. Eukaryotic cells also have a lot of organelles. And while they can sometimes live as single cells, when we talk about complex multicellular life, we're talking about eukaryotic. Eukaryotic cells, we call organisms with eukaryotic cells eukaryotic organisms. This is a simplified prokaryotic cell. And you can see it's got a couple of things. It's got a plasma membrane. So where is mine? So this is prokaryotic. The plasma membrane will sometimes be called the cell membrane. Basically means the same thing. It's a membrane that wraps around the cell. It's got DNA. It's got cytoplasm, ribosomes. Ribosomes are sites of protein manufacturing. And that's kind of it. Not really much else going on there. So it can do all of the basic things a cell can do. It can make ATP, it can make proteins and carbohydrates. But that's about it. Most bacteria consume other organisms for energy, so they eat living things or dead and decaying things. But there is one group of cells called cyanobacteria that can do photosynthesis. And we're going to see these guys are very important, and they're probably the origin of the chloroplast, which is an organelle in plant cells that's critical for photosynthesis. We're going to get to that probably next time. And this is a basic animal cell as an example of a eukaryotic cell. Remember when we looked at the pictures earlier and I said that's what a real cell looks like, and the drawings never quite convey how crowded and how much stuff there is? This is the kind of drawing I was talking about. This drawing is very good for learning about these sorts of things, but it's a model. It's not an accurate representation. So eukaryotic cells defined by the presence of a nucleus. And it also has all of these other organelles: Golgi, mitochondria, lysosomes and peroxisomes, endoplasmic reticulum, all kinds of things going on in here. Now, both types of cells have a plasma membrane. And it's basically made of the same kinds of stuff. Phospholipids, both types of cells have a cytoplasm. Cytoplasm just kind of refers to the liquid gel type material inside the cell. So it's a watery solution. It's thicker than water, so more like room temperature jelly or something. And both use ribosomes for protein synthesis. So you're going to see that in both prokaryotes and eukaryotes. Okay. What just happened there? Okay, something weird just happened on my computer. I don't know if you're seeing the same thing I'm seeing. I lost my screen. Okay. All right, I have another quick poll for you. It's just a one question poll to see how well everyone's paying attention. Oh, so far we're doing good. So far we're doing very good. All right, I'm going to end the poll. It looks like we've still got a few people left. Okay, that's a little better. So as a whole, you guys are right. The defining difference between prokaryotic and eukaryotic cells is the presence or absence of a nucleus. These other things kind of just follow along with that. The size has a lot to do with whether or not the cell has a nucleus. The structure of DNA, again, some of that is because of whether or not they have a nucleus, but that understanding why those things go together, well beyond the scope of this class. So I'm just going to tell you they do go together. All right. Great. So now I want to talk about the origin of the eukaryotic cell. So prokaryotic cells came first. We know this. We have fossil evidence for it. But at some point, something had to happen to a prokaryotic cell that allowed it to become a eukaryotic cell. And there's two major competing hypotheses here. One is that the nucleus formed, and then all the complexity of a eukaryotic cell followed. That's one idea. I think it's the less believed hypothesis. I think more people believe this second hypothesis. And that's that the mitochondria joined another cell and allowed more energy to be produced, which was needed for the added complexity of eukaryotic cells. And this is the one I like. I don't really like this one. So, a primitive prokaryotic cell formed a relationship with other prokaryotic cells that were what we call aerobic bacteria. Aerobic means using oxygen. Using oxygen to convert food materials, biological molecules, into cellular energy, you get a lot more energy than you do from anaerobic energy production. Anaerobic energy production gives you very, very little energy. So you've got some bacteria who can use oxygen, who can use that to make a surplus of energy. And they form this symbiotic relationship. And their host cell provides special structures for these bacteria. So it wraps them in a membrane so they're safe and protected. It provides them with nutrition. It protects them from potentially harmful other bacteria or other prokaryotes. And the host cell gets energy. And when it gets that energy, that means it can afford to make membrane that becomes the nucleus. It can afford to make various organelles that are specialized, but take more energy to function. It can get bigger, which takes more energy to function. So it can do all of these things because these aerobic bacteria evolve into mitochondria. Mitochondria came first. Plants went the extra step. They got a second symbiotic relationship with a cyanobacteria, one of those bacteria that can do photosynthesis. And those became the chloroplasts. We have evidence mitochondria and chloroplasts have DNA. So not all of your DNA is in your nucleus. And you only get your mitochondria from your mother. So technically, you get more of your genetic material from your mother than from your father. It's a little bit of a difference, but you do. Physically, they look similar to bacteria. They have an inner membrane. So remember, I mentioned that the host cell makes this outer membrane that's protective. That outer membrane looks like the kind of membrane we see in eukaryotes. The inner membrane looks like the kind of membrane you see in bacteria. So it's got different proteins in it. It's got different lipids in it, all of that kind of jazz. So they really do look like bacteria. This is called endosymbiotic theory of mitochondrial origins or chloroplast origins. Endo, inside, symbiotic, it's a symbiotic relationship. And that's how we believe that mitochondria originally arose and allowed the cells to become complex eukaryotic cells. How are we doing, folks? Anybody got any questions? Things that you don't understand? We've got one person responding, good. All right, everybody seems to be keeping up. Good. So I'm going to stop it here because the only thing left to do is kind of go through the structure of a eukaryotic cell. So this is an excellent place to stop. We should have plenty of time on Wednesday to go through the details of the inside of eukaryotic cells. And then, as I said, next week, exam, ecology, and then we're back in person, which I'm excited about, and I hope you are too. All right. In that case, I will see everybody next time. Feel free to email me if you decide you need me to explain something again, or if you have questions for the review session on Friday. Bye, everybody.
Bye. Bye. Have a great day.
Bye.
Oh, wait, I was going to ask if you got me for attendance.
Yeah, I got you. Don't worry.
Okay, thank you. Bye-bye.
Bye.